Hello, everyone, and welcome to Genome Webinars. I'm Ben Butkus, Editorial Director at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Digital PCR Method Development for Accurate Quantification of Copy Number Variations, and is sponsored by Stilla Technologies. Our speaker today is Dr. Alexandra Whale, Science Leader at the National Measurement Laboratory at LGC. Attendees may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Dr. Alexandra Whale.
Hello and welcome everyone to the webinar today. I am a science leader at the National Measurement Laboratory, and today I'm going to talk to you about digital PCR method development for accurate quantification of copy number variations. So just a brief overview so that you know what to expect. I'm just going to tell you what the National Measurement Laboratory, or NML, is, and then a very brief overview of digital PCR, with some background to copy number variations, and then what I've been doing to use digital PCR to measure the copy number variations. And I'm going to use the HER2 gene amplification in cancer as an example. I'm then going to finish up the webinar showing some data on how I use PCR to evaluate sources of bias within measurements, and then talk a bit about the future work, and then there'll be a Q&A session at the end. So the National Measurement Laboratory is part of the UK measurement strategy, and we're funded by BEIS, which is the Department for Business and Energy and Industrial Strategy. The NML is a designated institute for chemical and biomeasurements, and there are a number of measurement institutes in the UK that make up the full measurement strategy. Most countries in the world have some form of measurement laboratory, and we do most of our work in the inorganic area, organic area, and then molecular, digital, and cell analysis area. And we work across a number of sectors. We do research, where we work in collaboration with universities, hospitals, and industry. But we also perform calibration facilities, as well as producing reference materials and some training and consultancy. We also have a government chemist function, as well as being a national reference laboratory for a number of areas. So I work in the molecular and cell biology division, and we look at a number of sector areas. So we look at food and the environment. So we look at food authenticity and safety in this context. But a lot of the work we do in my particular group is advanced therapeutics. So we're looking at diagnostics. In a minute, my slides have skipped on. Please excuse me while my slides have reset. So we look in the diagnostic area. So we do a lot of work with infectious diseases or antimicrobial resistance, but chronic diseases, so things like cancer and neurodegenerative diseases. So I want to just give a little bit of a background to why we do the research we do. So we always think about measurement matters, and how can we be sure that the unit and the measurement that we're making is accurate. And this graph here just shows how important it is to be able to give confidence in the measurements. And in the context of DNA, it's sometimes a little bit abstract. So if I was to give you the example of the meter, if we hadn't defined how we change and think about the distance of the meter over the years, where we've evaluated the sources of bias and corrected them, then when we went to try and land on the moon, we would have missed the moon altogether. And so that in that context, in terms of the diagnostics, if we have someone and we are diagnosing them or monitoring their progress through a disease, we want to make sure that we're accurate, making over time, we can compare back to see whether they are responding well to a treatment or if they are in fact getting sicker. So the other things that we look at is feeding in to databases. So the JCTLM database holds the higher order reference materials and measurement procedures. So this is where we can go back to methods that are traceable to a unit. So in terms of DNA or chemical analysis, we might think of our SI unit as being the mole or the kilogram. And this is run by the BIPM, which is based in Paris. So the reason this is important is last year we were successful in submitting the first PCR-based method to measure nucleic acids. And we used the KRAS G12D mutation in this context because validating whether a tumor is KRAS wild type or with a mutation stratifies that patient into what medicine they would receive as part of their treatment. We also have ISO accreditation by UCAS for doing digital PCR measurements. And we feed into the ISO guidelines for routine applied methods, looking at other laboratories that need accreditation. And then we also have research guidelines. So the dMIQE guidelines that were published in 2013 are an example of this, on how can we produce publications in the more academic area that give reproducible material methods, but also other laboratories could replicate the findings. So I want to use this slide as by way of a disclaimer. Back in 2006, we were the first lab in the UK to have the Fluidigm Biomark. That was the first instrument onto the market. And then over the years, we've had a number of platforms for digital PCR come through our lab. With the latest one to be launched being Stilla NIQE system. So the data I'm going to show you today comes predominantly from the Fluidigm, the Bio-Rad QX200 and the Stilla system. So in terms of giving you the context in what I was saying earlier about having traceable measurements, I just wanted to start by putting up this slide here. And this was part of the publication that supported that JCTLM database submission, where we looked at measuring the KRAS G12D mutation, that's a single nucleotide variation, across the 5 different commercially available platforms at that time. And we can see that they're all very close in agreement. In fact, there's actually only 20% difference between the measurements. And while you may think that's quite a lot, these were actually all done without any calibration. We essentially put the material in the instrument, measured it, and then plotted the data here. So this was the ClinChem paper where this was published. But in the next month or so, we're looking to publish an update to the dMIQE guidelines. So that's dMIQE 2020. And of course, this we include the NIQE system from Stilla, which is now part of our lab. And this is just a comparison here between the QX200 from Bio-Rad and the NIQE system. And we can see that they're very, very close. And in fact, there's actually only about 2% difference between the measurements. So as expected, the digital instrument agrees closely, and then this is just a snapshot of the special report, so the dMIQE 2020 update. So the next few slides, I'm just going to give a very brief overview of how you perform a digital PCR. And if we think of a normal PCR reaction, in this example, real time, you have a 20 microliter reaction. And digital PCR, you essentially do the same. You assemble the reaction that contains the enzymes, the buffers, the template you need, but also the primers and probes that allow you to selectively amplify a particular sequence. But instead of putting this reaction into the cycle, you do an extra step. And this is where you split that sample into discrete partitions. It's often referred to as a limiting dilution. And the key here is that some of those partitions do not contain the templates. So in the schematic here, rather than having one 20 microliter reaction, I'm showing you 21 microliter reactions. And we can see that each of those black lines that represents a DNA molecule has gone into its own reaction, and some of them are empty. You then perform your PCR as normal. So if you have a probe-based chemistry or an evergreen chemistry, that happens as well with digital PCR. And then following that, you get a signal, so that fluorescent signal whenever you had that molecule to begin with. And each of those partitions is then read and then assigned as being positive, where you get fluorescence, or negative, where you just have a background fluorescence. And it's just looking at the proportion of this positive out of the total number that gives you the copy number concentration. you do need to apply a Poisson correction, because as the concentration of your template sample increases, the probability of you getting more than one molecule in the partition also increases, and the Poisson correction allows for that. Now, in reality, from this particular schema in the commercial into thousands to tens of thousands of sub-nanometer partitions. So at this point, I just want to go through some of the terminology that I'm going to use, just so you're aware of what I'm referring to. So we have the term partition. This is often given the symbol little n, and these are those individual subreactions within the digital PCR reaction. There's two main ways that this is done. There's either a chamber-based method where you have a prefabricated single-use consumable, and within that there are physical partitions that the digital reaction is subdivided into. The alternative is then the droplet-based, so this is where you generate water and oil emulsions to create those partitions. Positive partition, or K, so that refers to the partition that has a PCR amplification signal. And then Poisson, that I mentioned before, that's the mathematical correction to account for any of those positives that may have initially contained more than one molecule. And once you've applied the Poisson correction to the proportion of the positive partitions, you get what's called lambda, which is defined as the average number of copies per partition once you've corrected for the Poisson. Now, that Poisson distribution does have a number of requirements and assumptions. So the requirements are that it's talking about discrete molecules. So with PCR, we measure whole amplicons. You can't measure half a molecule. It's either present or absent. And then the probability of a given number of events occurring in a fixed partition. Essentially, what that means is we need the partitions to be of equal size. And then we also need a random distribution of those molecules. So that is, every single partition has an equal chance of having a partition in it, having a molecule in it, or not. Just popping up this little figure here, because I'm going to talk about this later on. This is an example of non-random distribution. So say we are interested in the blue target. If these are linked in the same molecule, when they go into that partition that's represented by the circles underneath, all three will go into one reaction. Now, even if we apply that Poisson distribution, we will under-quantify how many targets are there. So I like to think of digital PCR in its contribution to the field, and I feel there are four main areas that digital PCR can contribute to the molecular analysis. It's improving existing measurements. So we're looking at quantifying smaller fold changes. There are other methods that can do this. You can do this with real-time PCR or sequencing, but essentially digital PCR will allow you to quantify smaller fold changes. For those who are interested in minority targets fall in the same category here. We can measure smaller fractional abundances with digital PCR to say real-time PCR. We use digital PCR a lot in our lab to validate the pre-analytical processes. That is, we will use it to evaluate extraction efficiency. So look at different extraction kits or perhaps reverse transcription enzymes if we were looking at RNA. But we also use it to support existing measurements. So I talked about the traceability chain up to the SI unit, and say a lab has a real-time PCR machine, they don't have access to a digital machine, we could use digital PCR to value assign the calibrant materials that they then use. And then, of course, you can make totally novel measurements with digital PCR. And for this, I'm talking about things like higher order multiplexing or drop-off assays that are incredibly difficult to do with other technologies. So I now want to talk about copy number variations and digital PCR. So just as way of an introduction to copy number variations, it's essentially where a section of the genome is repeated or deleted. These can be structural variations where you get duplication or deletion, and they can vary within individuals. So they're not necessarily associated with a disease state. And they think about 5 to 10% of the human genome can be classified as copy number variations. And really the access now of next generation sequencing has allowed them to interrogate the genomes at a much bigger, greater level than they used to be able to. So there's two main copy number variation types. They have the short repeats. So these are the 2 or 3 nucleotide repeats. These occur in humans, but can often lead to a disease state if the repeats reach a certain threshold. And the example here is Huntington's disease. Once that trinucleotide repeat gets to a certain level, you get the buildup of protein in the cell, which actually causes the cell to have a pathogenic effect on the human. You then have the long repeats, which tends to be more what people think of when they talk about measuring CNVs. This is where you could have an entire gene repeated. And a really nice modern example of very recent human evolutionary adaptation is the alpha amylase 1 and how the copy number is related to diet. But there's also defined hotspots in the genome that are more predisposed to having segmental duplications. They can be tandem or interspersed, and all cause an increased chance of chromosomal rearrangements. In general, when people talk about CNVs, they're not talking about polysomy, although of course polysomy and monosomy are copy number variations. They're just at a chromosome level. And of course, an example here would be Down syndrome. So the areas that these are looked at are predominantly in the disease, so the health versus disease area, things like precision medicine, so you're looking at the copy number variations of disease states, and this will be the HER2 example that I come on to. But also in prenatal diagnosis, there's now a really nice number of methods out there that can start to look at chromosome disorders before a baby is born. And then I just hear also the area of engineering or synthetic biology, the idea that if you transfect or transform a cell with, say, a plasmid, you could start to look at how many copies are actually integrated in that genome. That would be a copy number variation. So the example study in this webinar is detecting the HER2 gene amplification. So a proportion of breast and gastric cancers are associated with the amplification of this gene. And the reason this is important is because they have a specific treatment for HER2 positive tumours. The patients are stratified, and if they have this HER2 amplification, they are given Herceptin. And it's really important that patients who don't have HER2 positive tumours, so that HER2 is not amplified, are not given this treatment, because although it's very effective for a HER2 positive tumour, it does nothing for someone with a negative tumour, and in fact can have some quite nasty side effects. So the gold standard method for detecting HER2 amplification is using fluorescent in situ hybridization, or FISH. And I've got an example here that I've taken from the Herceptin website of a normal cell at the top with a HER2 amplified cells at the bottom. So each cell is a blue circle with a red dots representing the HER2 gene, and then centromere 17 as the green gene. And these cells get scored depending on the ratio between the HER2 and the Sen17. So less than 1.8 is negative, so that patient would not receive Herceptin. And then 2.2 or above, that patient would be eligible for Herceptin. And then we have the borderline cases. An alternative method is histochemistry, or IHC, and this is where we look at the protein. So this is the growth factor that's found on the cell surface, and the darker the staining, the more HER2 protein there is in the cell. And so at the top, I've got, again, a normal cell staining with HER2 amplified cell staining at the bottom. And there's been a lot of work over the last 10 to 15 years in trying to develop a method for determining the HER2 status. The reason for this, both FISH and immunohistochemistry, are quite time-consuming. They require experienced personnel. So why would we want to use a molecular method, and how would we go about that? Well, as soon as we go into the molecular state, we lose that cellular context. We have a tube with DNA in it. So we're going to start looking at the fold changes. So similar to what we see with the FISH, where we look at a HER2 to reference gene. Now, we need that reference gene because that's essentially a surrogate for our cell number. We can no longer see the cells count how many HER2 molecules there are. So what we have is a relative measurement. The PCR-based methods are a really good candidate for this. They're generally easy to perform. You can design assays that are specific for that target and reference. But there does need to be selection. The design of that reference assay is really important because HER2 amplification is not the same as polysomy chromosome 17, that is the chromosome that HER2 sits on. So why would we use digital PCR? Well, it gives you an absolute quantification. If you remember back to the cartoon I gave you, we count the number of positive reactions. We don't have to convert a fluorescent output into a copy number. So we don't need that calibration curve in order to get that quantification. We have this lovely digital output. We just count the proportion of positive partitions. And this makes it suitable for a reference measurement procedure, because it's not tied in to, say, UV absorbance or any other method. It's down to the SI unit of counting, which also means it's suitable for trace detection. So if we're looking at those sort of borderline cases of HER2 amplification, we're likely to have a method here that could detect those small, subtle changes. It's also highly precise because we're looking at the mathematical correction. We can measure very small, but also remember that slide I showed you right at the beginning with the KRAS SNP detection, so a single nucleotide variation. We are able to get high reproducibility between the different digital platforms, and when we do interlab studies, we also see high reproducibility. It's also less susceptible to inhibitors or matrix effects because we're looking at that endpoint and it's the presence or absence. We don't have to wait and find the point that it reaches that exponential amplification and set a threshold that way like you would with real-time PCR. So back in 2012, when we had the Fluidigm instrument, we started looking into this and we looked at the HER2 status of a number of breast cancer cell lines. This is a figure I've taken from that paper. And with the 3 different cell lines here compared to normal female genomic DNA, we got very good concordance between the qPCR and the digital PCR and the karyotype and the HER2 status, of those particular cancer cell lines. So the MCF7s are monosomy for chromosome 17. So we can see we had a less 0.44 ratio. And with the T47D that has a doubling of the HER2, again, we got a really good concordance. For the SKBR line, HER2 is amplified in cis. This means you get 2 or more linked HER2 targets. So if you remember back to that assumption slide when I was first introducing the Poisson, that means if we have HER2 amplified in cis, so they're in tandem, they're on the same molecule, they will be segregated into one partition. There will not be independent segregation. And in this particular example, we can see that you get an under-quantification using digital PCR by about 20%. Real-time PCR, of course, doesn't have this problem because it's a big mix. It's a single 20 microliter reaction where we're just looking at the overall increase of fluorescence in the reaction. Now, the diagnosis from this, if it was a biopsy, would be correct. We're clearly over that HER2 ratio of 2.2. So why does it matter in this case that digital PCR has under-quantified compared to real-time? Well, this was expanded in about 2013, where they looked at the HER2 status of tissue biopsies in FFPE tissue. So FFPE tissue is pretty damaged. The fixation methods that they use can cause quite a high level of fragmentation of your samples. And they used here a number of samples where they looked at the HER2 status using digital PCR and compared it to the immunohistochemistry. So that for this, they used the Bio-Rad QX100, which was this droplet-based instrument onto the market, and they used a centromeric on chromosome 17 as the reference, and they compared this also to fish. And what you can see is that digital PCR showed good concordance with immunohistochemistry. There isn't fish data for every single one of these samples. But the challenge was in the classification of those samples that fall in the borderline range. So the threshold that they have across here is the upper limit of that borderline. So it's sitting at about 2.2. So this was a nice proof of principle that digital PCR could measure as well as immunohistochemistry or FISH. So I just want to change the rhythm here slightly and just talk briefly about minimally invasive or non-invasive diagnostics, because this is where the HER2 field started having publications in. If we think in terms of cancer diagnostics, you initially have that genotype, often from that initial biopsy, and tumors can be highly heterogeneous. And also they know that amplifications of HER2 can be acquired and lost throughout either the tumor progression or in response to treatment. And in fact, the secondary tumors or the metastatic tumors can have different mutations or different levels of amplifications, even in the absence of the primary tumor. And so the idea of using cell-free DNA was first suggested, and this would allow continuous monitoring. You could potentially assess the full heterogeneity of a tumor. But also, you could only analyze this using a molecular method. So the DNA is just found in the circulating system, and it's not in cells. So something like FISH or immunohistochemistry would not be able to look at how many HER2 copies you have. Now, very briefly, what is cell-free DNA? Well, it's the non-cellular DNA in the body fluids. It's non-cellular by nature rather than the extraction. There's lots of sources. I've given two examples here of the blood or urine. And it's comparatively easy to access. You know, you could provide a blood sample or a urine on a reasonably regular basis. That wouldn't be possible with an actual tissue biopsy. It comes from normal cells, but also from tumor cells. It's a relatively low, but there's a large range that you find in the blood plasma, and there's natural variation between individuals that will change over the course of a day, whether you're tired, stress, if you've just done exercise. And it comes either from yourself, so from your normal cells, or from, say, a tumor or in an immune response, but also non-self. So the fetus will contribute to the cell-free DNA, or the virus or pathogens, or in fact an organ transplant. So how does this relate to copy number variation measurements? Well, if we think of cell-free DNA, it's naked within the blood. It's often highly fragmented. It sits around about the 200 base pair fragments, which means you can get molecular dropout. Now, molecular dropout is the term we use for anything where the target is present, yet for some reason it doesn't amplify. So if you have a cut site or fragmentation event that happens between your two primers, you will not get a PCR amplification. So you will under-quantify the number of targets that are there. Cell-free DNA is also quite low in concentration, but also it's only a proportion of that cell-free DNA is derived from, say, the tumor or the fetus or an organ transplant. And there's a really nice graph here or table that Stiller produced in one of their talks, where if we look at the copy number variation along the top and then the percentage that it contributes to the cell-free DNA down the side, we can see why measuring a small copy number variation would be of use. So say our copy number variation is 5, but only 5% of our breast cancer tumor contributes to the cell-free DNA. Therefore, we actually need to be measuring a 1.2 ratio rather than a 5 ratio. And then I mentioned just before, but the cell-free DNA is not just looking at cancer number variations in it's also looking at mutation monitoring. And in fact, they use EGFR SNP monitoring with digital PCR. So in order to start testing out what we could measure with digital PCR, we developed a copy number variation using a cell line genomic DNA. And this was back in the 2012 paper, where we generated different ratios and measured them with digital PCR. And we found that our limit was about 1.17. This was using the Fluidigm, where the reaction partitioning is very low. It's about 770. We used 8-pooled reactions, so we had an effective partition number of 6,160. Recently, after we published this back in 2015, the Institute of Cancer Research also published HER2 amplification in plasma, where they were looking at 46 metastatic breast cancer patients. So again, these have been determined by SISH, which is section in situ hybridization or immunohistochemistry. They also did a large amount of work in trying to establish a really good reference region in chromosome 17, and they looked at a cohort of patients with HER2 amplified or non-amplified to identify an optimal chromosome copy number reference region, which is called EFTUD2. They then did a model called the sequential probability ratio test model, and I've put the reference for that paper down at the bottom for those who are interested, to essentially evaluate their system. They used the Bio-Rad QX100, and they had really good correlation with pretty good sensitivity of 80% and specificity of 70%. So we're looking at improving existing measurements. How small can we go? When we were looking with the fluidine, we got down to about 1.17, but can we actually go further, and can we develop a model for predicting it? We want the template concentration to be taken into consideration because we might be using cell-free DNA or tissue biopsies. And then could we predict what will be the lowest? And we developed this mathematical model. And as you can see along the bottom, we've got the copy number variation we might want to measure with the number of partitions required. Now, of course, this is slightly outdated because we only went up to 10,000 partitions. And we now know that digital instruments that can go far higher than this. We also show template concentration. Of course, you can increase your resolution. And so it was the partition number that seemed to have the biggest effect on increasing the ability to measure small numbers. Back in 2018, Stiller presented this rather nice graph where they used the same method we did back in 2012, but they used their system where they have about 25,000 partitions per reaction. And you can see that if you were to look at a ratio of less than 1.1, you would only need 3 reactions, even when your lambda is really quite low. In this example, the blue line is about 0.3. So we started to develop at the NML a copy number model. So we took 2 plasmids and we thought we would generate incredibly small copy number changes where we kept the constant reference and varied the target. We used a FAM probe for the target and a hex for the reference. And this is an example of the output. So this is called a 2D or a 2-color scatter plot. What we have along the bottom is the level of fluorescence for the reference. And on the y-axis, the FAM probe or that target. And we can see that there are 4 clusters here. Each of those little dots represents 1 partition. So we can suddenly see the scale of the number of partitions and the resolution here. They're color-coded based on what molecules they contain. So we've got our empties that are gray, just the reference is green, just the target is blue, and then a partition that contained both molecules is the cyan color. So we generated a number of ratios between 1 and 2, and we used the Bio-Rad QX200 and the Stiller MIQE system to start measuring them. And what we have here is a combination of data from those platforms to show you how that increase in the partition number has increased that resolution. And this is just reproducing the data we had from the fluidine biomark, where we only had 770 partitions, just to show you how small those error bars are now because we've increased the precision, because we've increased the number of partitions. And of course, because we could measure between 1 and 1.2 with quite easily, we then generated a number of other ratios between 1 and 1.2. And we looked at the variation across different days and different operators. So we've got quite large error bars here, but we're trying to pull in all the measurement uncertainty that we might get in these examples. If you were to go into a laboratory hospital, you can't always expect the same operator to be there. So having a robust method that can work and give you reproducible results over different days and with different operators is really key here. This data shows that really nicely. We could easily measure that 1.1 ratio, but also the 1.05 is pretty good. Now, actually, the problem we had was our ability to make the small ratios. We used gravimetric dilution, so rather than just using the pipette, we were weighing out our samples to try and get an uncertainty. And the error bar that I've got going across the screen is actually uncertainty associated with the scales. So you can see quite clearly why we couldn't get any lower than 1.05. We just couldn't physically make the material in order to assess digital PCR. So digital PCR broke our ability to pipette accurately. So I want to finish with the factors that impact on copy number measurements. Now, because we're a measurement laboratory, we're interested in all the little things that might be causing bias. What would pull our measurement away from that true value? And so I want to focus on linked targets. So there's a number of things that could be impacting. We've talked about the digital PCR method. By that, I mean the partition number, perhaps that partition volume variance, or the reagents. I haven't talked about those today. But if you use polymerases, perhaps that impacts on your ability and the copy number variation, and also the template concentration that I've talked about. The digital PCR assay is, of course, important, and there's a nice application note from Stiller that shows you how you can use their 3-color system to actually account for when your HER2 copy number might officially elevated just because there's chromosome 17 polysomy in that sample rather than HER2 amplification. And then the detection chemistry we did as part of the JCTLM submission for the KRAS work. We looked at a number of different ways generating that fluorescent signal. So I'm going to talk mainly about linkage, but also touch on the genomic DNA versus plasmid fragmented DNA, so the DNA type. So when we have those linked genes, we can get this under quantification. This is just a schematic on the right to show the reference gene in green, and then we've got 3 blue targets amplified in cis. And I like to think about how would this be measured if we were to look at a digital reaction. So what I've done underneath is just sort of make a cartoon of how I think this would partition. So we have 4 possible scenarios. We've got the empty droplets, the ones that just contain our blue target or our green reference, but also the droplets that might contain both copies. Now, the problem with this is we would end up with a ratio of 1. So even if we apply a Poisson distribution, we would still under-quantify what is there. So really what we want to do is separate those targets before we do the partition, and then we would actually measure the correct ratio. So there's 2 ways you can do this. You could do some sort of pre-amplification on a sequence-specific stage. We've looked at this in the past. We published in about 2013, looking at low concentration detection with digital PCR. And essentially, if you do a pre-amplification, you can generate a significant bias of 2 or more fold between your different targets. So this is not appropriate for this particular scenario. So the other way is the fragmentation. You can do this enzymatically or shearing. So the advantage of an enzymatic is you'd have a more control, but of course you have to know the sequence and know that your enzyme will cut between your targets and it won't cut your amplicon. Shearing is a bit more random, but it would shear in a sequence-independent manner. And of course, whatever method you use, it's got to be independent of no mutations or methylation status. So I'm going to talk about the enzymatic work that we've been doing. So we developed a model to detect the loss of linkage, which was essential plasmid that we would then digest. But the advantage of this model is, say we only partially digested. So in this particular example I've just given you here, we've only separated one of those targets off. We wanted to be able to look at this kind of scenario and recognize it, because of course, if we think we are separating all our linked targets, but not separating all of them, then we're going to have a biased measurement and we're going to be unaware that it's biased. So we need a well-characterized material. We need to be able to know when we've removed that linkage, and we need to know what the true ratio is. So when we're measuring something different, we know that we're measuring something as a response to that separation or in-separation. We want a low template complexity. We don't want to start with genomic DNA or hetero mixed cell populations where it's suddenly a bit more complicated. Again, we won't necessarily know exactly what that ratio is. And then we need some sort of metric to determine linkage. So with digital PCR, because we have this partitioning, we can actually predict where we would expect all our molecules. If we were to measure 10,000 droplets and we'd have a lambda where we would expect 8,000 of those to be positive, we can actually work out when we're not getting 8,000 or if we're getting that different change in distribution of single positives or double positives. So we want to be able to compare this to the distribution for unlinked targets. I'm going to show you some slides on exactly what I mean by that. And then we needed a platform that can identify the different targets. So I just want to introduce you to the ADH plasmid quickly, which I mentioned earlier. This is one of our in-house non-human reference materials, and we use this a lot because we can spike it into various human-based samples, and we know it won't interfere with the copy numbers of the humans. It's a linearized plasmid, and it contains a sequence of the alcohol dehydrogenase gene from Arabidopsis thaliana. We have 3 targets in cis. We call these the ADH targets. They're at a ratio of 1 to 1 to 1. So we know what the ratio is. And then we can design assays for each of those called the alpha, beta, and delta assay. Now what I've got here is just the various coloured lines, blue, green, and red, to associate the different probe colour. And for the rest of this talk, when I use the 3-colour instrument, I'll be using the alpha on the red channel with a Psi5, the beta on a blue channel, and the delta on the green channel with a hex. The advantage of this plasmid is it's got a really well-characterized digestion scheme. We've digested it with pretty much every enzyme that has a restriction site within the ADH sequence, so we know exactly what we need to do to separate those targets. And then actually what's really nice is we want to go up that level of complexity. This sequence is exactly the same that's found in DNA. So our loss of linkage model, we need to target 3 different targets. So these are on 3 different colors. And if we look at how those would segregate into the different partitions, so the circles are a partition, we get them on their own or we get none. And then of course we get all the different double combinations and then a triple. Now, when we use a 3-colour MIQE system, these get given a pseudo colour. So purple is red and blue, yellow is green and red, and then the cyan colour that I have shown you in a previous plot is when we get both the beta and the delta. And then, of course, we get a triple as well. And we get an image that looks like this. So this is very similar to the 2-colour plot I showed you in the past, except we've got 3 axes. So we've got the red axis, that's the ADH alpha, the blue, that is level on the lower part off to the left is then hex goes up. All coloured by how I showed you before. And then what we can do is we can compare it to linked molecules. Except we would expect only 2 outcomes. Everything's linked, so they're only going to be that triple positive or they'll be the empties. And this is the kind of output we get. We can apply the numbers to it based on where those molecules are and how they've been pulled out of the correct cluster that they should be in, should they have been independently segregated in the partitions. So I want to show you a really nice example, and this is one of the things I really like about digital PCR, because you get this really visual output. You can actually start to see where things might be going wrong or where something unexpected has happened. So what I've got here is the alpha, beta, and delta stacked on top of one another, and then each of the reactions have a different number of unit of the enzyme added to them. So the first one has no enzyme, and then 6, 12, 24, 48, 96. And essentially I kept adding enzyme until the reaction didn't work anymore. This concentration is what I call the equal partition concentration. That is, if they were completely unlinked, we'd have an equal number of those 8 partitions. So it makes it really easy to see when anything is distorted, the number of partitions in each cluster is distorted. So there's 2 main things you can see from this. Firstly, you add too much enzyme and it affects your fluorescence. And we can see that the 96 units, which of course is a colossal amount of enzyme to add, has really disrupted that reaction. It's almost knocked out the ADH alpha, so you would be underquanting that. But you can see a drop in the final fluorescence of the beta and the delta as well. So it also tells you that it's different for each assay. So you can't even say, don't add this amount of enzyme enzyme because your digital assay in this way, it's assay specific. And then, of course, the big one is when we look at that ADH alpha assay. We can see there's two populations. And why? But when we get to 48, we can see that there's only one population. So what is going on? We can see that there's predominantly one population when we add no enzyme, but two when we start adding it. If we just look in on that, and I just expand it up a little bit, what you can do with the software is you can actually pseudo-color those droplets based on where they are in that 3D plot. The grey ones that are at the forefront are the triples. The purples have the alpha and the beta. And what's really interesting about this is that second population of reds that is higher, they only contain either purple or grey. So the hypothesis here is that the alpha and the beta have remained linked for some reason because there's a disproportionate number of purple droplets compared to blue or red. And then, of course, there's a probability that triple positive alongside a delta, and that maths followed. So what we had here was like a really nice way of visually showing we hadn't added enough enzyme in order to efficiently separate the alpha and the beta. Let's put up this example because, of course, we tend to think mainly in copy numbers, and we can see that even with those enzymes, even with that endpoint fluorescence, we were still getting pretty good correlation in how many copies. We're expecting about 8,000. And of course, the 96, we've massively underquantified that ADH alpha. It was horribly compromised by the amount of enzyme. But between the assays, so alpha, beta, delta, we only had a CV of 2.5, and between the enzymes, 2.8. So this gives you an idea of how precise digital PCR is. And we've looked at other factors as well as this, like other enzymes and the different digital PCR platforms. So how does this relate to HER2 amplification? And we need to move into the genomic DNA. And this is where I'm just putting up some of the mile-marker assays, which were published by Regan et al. In 2015. And we've got a duplex of assays that are 33 kb apart or 210 kb apart. And just looking at normal human genomic DNA, we'd expect a ratio of 1 to 1. But when they're 33 kb apart, we're actually getting a linkage of 56%. So we're getting a disproportionate number of doubles. And then, as you would expect, when we get to 210 kb apart, we're pretty much back down at where we would expect two completely independent assays to be. So we can see with these scatter plots a clear difference in the populations. But the ratio between those targets is comparable. So what does this mean? So if we think about the ADH plasmid, because we designed 3 different assays, we could actually start to look at that residual linkage that happened. Now, if we had a HER2 assay, we wouldn't be able to design different HER2 assays. We'd have one HER2 assay, but it would be trying to look at whether the different HER2 targets had been removed or not. But if we look at what we had with that mile marker assay, where we actually had 50% linkage, even though they were relatively far apart, 33 kb, we would still start to underquant. So this is really highlighting that the problem here is when you have that residual linkage. And while we might be looking at small changes, actually we could be looking at a 50% underquant. So what next? Well, there's a 6-color instrument that's due to come out, so we could start looking at more targets. You know, maybe we could have a target 33 kb, 60, 120. Let's start to see where we start losing that linkage, because we can start feeding that into our HER2 data and start looking at the distance between those targets. Then the other thing we're very interested in is this fragmentation. So if you remember the one that wasn't digested at all, we were expecting everything to be linked, we actually saw some fragmentation. Now, we often observe this. We had multiple plasmids, different sources using different digital PCR platforms. We often see it sits around that 3%. The reason this is important is because those reference materials that might be used for calibration, to calibrate other materials, or to try and get traceability up to the SI, they're often plasmid materials. So this would be an important bias to characterize. Maybe the mathematical assumptions are wrong. The maths I've used to determine the linkage, maybe it's wrong. Except because we've got digital PCR, we can see those droplets. We can see those clusters and those partitions. So we know that there is some separation. And then, of course, there's molecular dropout. As we get more and more complex with more and more assays in there, perhaps there's more interactions between the different assays. Perhaps in this case, the assays are compromising one another. So one assay is working and another one isn't. And then the other thing we want to look at is perhaps that partitioning step is causing some sort of fragmentation. When you look at the chamber-based methods or the droplet-based methods, at some point the reaction goes under high pressure. Perhaps somehow we're causing fragmentation. I suspect it's a combination of all of the above that is contributing to that 8% and that 3%. So because of time, I'm actually going to skip through this very quickly. This is actually published data in PLOS One paper back in 2013. It's predominantly using the fluidine biomark system to look at molecular dropout. Now, one of the things we want to look at is now we have a much higher partition instrument. Let's start looking at how the concentration of the genomic DNA or the plasmid can impact onto that molecular dropout. So in summary of this webinar, we're interested in defining those sources of uncertainty and bias. And in this particular example, we've looked at copy number concentration, copy number variation estimates. And it's the small uncertainties in many of those parameters that could add up to a large uncertainty. So you end up with a fully biased measurement that is not the same as your true measurement. Digital PCR is an appropriate molecular method to measure copy number variations. The high precision allows us to look at small fold changes. There's a large number of partitions, so we can increase that resolution and precision. You can also use it. It is appropriate to use over fish or immunohistochemistry because the process time is shorter. You don't have to rely on the operator expertise. We have some lovely interlab studies now that we've published in collaboration with other measurement institutes around the world. But also, you can start to look at whether that hERT amplification has come from chromosome 7 polysomy or is genuine amplification. And then I just showed you towards the end just how I like to use digital PCR to look at different aspects of the molecular method. So I looked at the loss of linkage model, but also you could apply this to other applications that use enzymes. So we could look at Cas9 enzymes or anything else that might cut the DNA. And so the future work is really just to publish this, looking at the 3-color digital model with the ADH plasmid, refine that mathematical model, and expand the linkage model to look at genomic DNA. So start to look at the material, or perhaps cell-free DNA, and how that would impact. And there is actually some materials out there for HER2 copy number. So NIST produced this back in 2016 using different genomic DNA cell lines to be used for either calibration. They've used it in a really nice study where they've evaluated different NGS methods. And also as part of the international metrology community, the CCQM, so this is part of the BIPM, the Nucleic Acid Working Group. I'm very sorry, I've just thrown 3 acronyms at you. Essentially, these are the international metrology labs, and we all like to compare can get in order to start asking the questions about traceability. And there's we're looking at copy number variations in the interlab comparison for the measurement. So I'd just like to finish with the members of my team, Anna and Dilek, who did a lot of this work, Goin, who worked with me on the KRAS work, but also Jim, Helen, Alison, Carol, and Alison, who've been instrumental in guiding the science within the lab. And for Stilla for inviting me to do this webinar. And I will end there, and we do just about have time for some questions, I think.
Thank you, Alexandra. As a reminder to webinar participants, if you have any questions, please type them into the Q&A box into the control panel. We'd also like to ask attendees to take a moment after the webinar ends to take our exit survey and give us your feedback. We'll now begin the Q&A portion of the webinar. The first question: for infectious disease detection, such as for the coronavirus, are there major advantages of digital PCR over conventional qPCR for instance, higher sensitivity or accuracy?
So digital PCR has been used to look at viral load monitoring over time in response to treatment. The main advantages that digital PCR would have over something like qPCR is that you wouldn't need to develop reference materials, so plasmids. You could go straight in and measure directly with digital PCR. However, what we've found in the viral community is that qPCR assays are working great. It's just the value assignment of their calibrant. So if you use a nanodrop or a UV spec method, you can be out by about a third. And so we've done some nice work where we have value assigned using digital PCR plasmids or cell lines that they use for viral load monitoring using digital PCR. And so I think in terms of the coronavirus, to suggest that digital PCR would be the best method at this point wouldn't necessarily help with disease detection, because a lot of labs, I would perhaps say most, have a qPCR instrument. However, they don't yet have a digital. And so I think it would be more beneficial for us to use digital to value assign calibrants rather than directly measure.
Thank you. The next question, how long is the runtime of digital PCR versus qPCR?
So the main difference is that partitioning. So you can set up digital PCR reactions in 96 well plates, in 8 well strip tubes, and then you have to have this partitioning step. Now, each of those commercial platforms has a different way of performing that partitioning. Some of them have more hands-on time than others, but ultimately it takes, sort of sometime between 40 minutes is probably the shortest time, up to about an hour. But I mean, it can depend on the operator. But I would say it's more the, so with the Stiller system, for example, it's all automated. You pipette into the consumable and then it's part of the cycling. Whilst with the QX200, it's a step before, but then the cycling takes less time. So that is the main time difference. And then, of course, you've got to read the partitions at the end. So it's not done in real time. You have that endpoint fluorescent and then processing the data. So I wouldn't say in total it doubles the qPCR time, but that is roughly what we would be looking at.
Thank you. The next question is a two-part question. Could you please provide your opinion of single-cell sequencing versus digital PCR? And are there methods for transposase-accessible chromatin digital PCR or reverse transcriptase digital PCR for the transcriptome?
So the main difference between the sequencing and the digital is, of course, with digital PCR, you need to design assays. So you need to go in knowing what you're looking for. And because you need to design those assays, and you would need to have a level of multiplexing, ultimately you can look at fewer targets. So with a single cell sequencing, you can look at the entire genome, whilst digital PCR would be more targeted. So while, of course, with the single cell, you can do more targeted methods. This would be the same for both measuring DNA and gene expression, so measuring RNA transcripts. It depends what you're looking for. In the context of HER2, you have a very specific target that you're looking for. If you're looking at changes in gene expression, then the larger number of targets, then the more information you would get. The second part of the question, transposase accessible. By that, do you mean looking like I'm not sure what you mean by that. In terms of the RT-dPCR for transcriptome, there are a number of RNA-seq methods for single-cell analysis. With digital PCR, you can do the reverse transcription. Today, I talked almost entirely about plasmid and genomic DNA, but you can do one step reverse transcription, digital PCR, or you can do it as a two-step. But again, it comes down to the number of targets that you're interested in measuring between different samples or in response to different stimuli.
Thank you. The next question. Would you consider the method you described today quantitative or qualitative in terms of CLSI guidelines?
Having thrown a lot of acronyms out, CLSI is what? Do I know it's something else? Can I come back to that question? Just my colleague is just going to quickly check.
Yes, it's Clinical and Laboratory Standards Institute.
Okay, sure. So it's predominantly, it's quantitative in the sense that we can measure small changes. We can measure them reproducibly. We can look at limit of detection, limit of quantification with them. In terms of the HER2 gene amplification, it's essentially yes or no is what you need the output to be. So I would say at this stage it's quantitative, but we haven't got anywhere near the rigorous levels of assay testing and development that would meet CLSI guidelines at this point.
Thank you. Unfortunately, that's all the time we have today for questions.
Sorry, I overran a bit.
We'd like to thank Dr. Alexandra Whale and our sponsor, Stilla Technologies. If we didn't have time to get to your questions, we will try to follow up with our expert.
Yeah, that's
As a reminder, please look out for the survey after you log out to provide your feedback. Thank you for joining us for this Genome webinar.